Hello, my name is Harper Van Steenhouse, and today I'd like to share with you an introduction to the TempO Seq gene expression assay by BioSpyder. Additional information and publications are available on our website at biospyder.com, and if you have any specific questions, please don't hesitate to email us at info@biospyder.com. The TempO Seq assay has been designed specifically for very efficient profiling of molecular signatures from a variety of inputs. As I'll share with you in this presentation, it's highly flexible to the number of genes you want to look at, from just a few to tens of thousands in the case of the whole transcriptome, and is equally amenable to purified RNA and nucleic acids as it is to crude lysates. TempO Seq itself is an inherently targeted expression assay. While it's targeted, this does not mean that it is limited in any sense. We are able to multiplex hundreds to thousands of genes up to the whole transcriptome, as I mentioned. In addition to specific isoforms, we can look at fusion genes, expressed coding SNPs, or other mutations in a variety of custom designed panels. As I mentioned, the assay is very easy to use in that you can perform it on crude lysates in addition to RNA purified and extracted from a variety of materials. This is because we do not go through a cDNA synthesis step. There's no reverse transcriptase activity required. And so whether you have cells in culture, fresh tissue, frozen tissue, or samples, in fact, that have been fixed in a variety of methods, including FFPE, these materials can be lysed using the TempO Seq lysis buffer, and the assay performed directly on those without additional preparatory steps. In addition, I'll show you that the bioinformatic analysis is simplified. This is because the targeted probes in the assay do most of this work for you and simplify the analysis downstream. While we've simplified a lot of these approaches and made the assay more cost-effective and efficient to run, the accuracy and sensitivity are extremely high. By virtue of not having a cDNA synthesis step, nor any preamp steps, we're able to get down to very low input amounts, whether it be because of degradation, because of limited amounts in the case of highly valuable tissue samples that are limited, or in the case of single cells or very small populations of cells. At the same time, the assay is amenable to very high throughput processing. Manual processing allows running 96 well plates, anywhere from 2, 3, 4 plates at a time, and these can be conducted well within a working day. Furthermore, the assay is readily automatable to expand to larger scale screens. This can be performed on essentially any liquid handling robot, and we have barcoded primers that support this high level of multiplexing on sequencers. In addition to the flexibility that I've described above, as I said, it applies equally to FFPE tissue and single cell applications, which we can discuss at a later time. Importantly, the assay is, as I said, highly flexible, and you're able to perform it for a number of different applications, including toxicology, drug discovery, oncology and immuno-oncology, biomarker discovery, a whole host of translational research, and as well, we're working on the diagnostic implementation of the assay itself. The content can span across any number of genes, from whole transcriptomes to defined subsets of the transcriptome, in fact, customizable for your particular experiments, whether it be a subset of the transcriptome or any species or any variant or any gene isoform you'd like to look at. The same assay works on all variety of sample types, including cell lysates, purified RNA, FFPE tissue, and getting into specialized applications such as liquid biopsy and single cells. So with that, I'd like to share with you the principles of the assay. As you see in this diagram here, typically we start with an input in a 96-well format, although this can be also performed in tubes if you have fewer samples. This can be cells in culture. It can be tissue that you've collected off of section slides, or it can be purified RNA. The first step is to add TempO Seq lysis buffer. This both allows access to the RNA as well as normalizes all of the salt concentrations in the input material. This is important because, as you'll see going forward, the assay is an add-only assay. There are no purification steps, there are no buffer exchanges, there are no bead-based cleanup steps, and so the material at the beginning remains throughout the assay. You'll take a very small amount of this lysate and add it to the assay plate. This 2 microliter transfer typically targets about 1,000 cells. However, as we can share, the assay works down to very small input amounts as well. To that 2 microliters of crude lysate, detector oligos are added. The detector oligos are what we term the targeting reagent in this case. The blue molecule here, the blue line, indicates the RNA target, and the detector oligos are depicted in green and red and green and yellow. Detector oligos comprise a pair of oligos that target a specific gene, and they will then hybridize immediately adjacent to each other to the appropriate target. Each of the steps following are performed in the 96-well assay plate and involve the addition of reagents that come in our kit, followed by incubation steps, typically in a thermocycler, but can also be performed in an oven. The first reagent step and second reagent step depicted here provide the specificity for the assay and the very low background that you'll see in the data. The specificity is driven based on the requirement that 2 oligos hybridize immediately adjacent to each other in order to be ligated in this step. Furthermore, background signal is reduced in this step by addition of a reagent that reduces the complexity and knocks down that potential off-target hybridization. At the end of these steps, you have a ligated molecule that is essentially barcoded in this green line, indicating the gene target that the detector oligos were designed against. The number of molecules of this barcode that are present in the assay product are in direct proportion to the number of molecules that were present for that target in the input material. At this point, we are taking advantage of these red and yellow universal priming sites that are used in a cycling reaction that incorporates tags that include index 1 and index 2, so that they can be combined and multiplexed on a sequencer, as well as the sequencing adapters that are required for the sequencing reaction on the sequencing instrument. This step can be performed in a qPCR instrument so that you can generate real-time signals as an internal QC, but this is not required. At this point, you have 96 samples, each of which have gone through the TempO Seq reaction, each of which are uniquely indexed, and so they can be combined into a single pool, then concentrated using a single spin column that concentrates and cleans this up and results in a ready-to-sequence library. This library is then either sent off to a sequencing provider for 50 base or 75 base single read dual index sequencing, or it can be performed on an instrument that you have running in your own lab. The sequencer outputs FASTQ files that are demultiplexed and therefore separate for each of the samples input. Once you have these files, the bioinformatics steps are extremely easy. Each of those FASTQ files that represent the raw data from each of the samples are, in essence, barcodes that indicate the presence of each of the molecules of, in this case, RNA that were targeted by the assay. The alignment step is greatly simplified compared to other methods because you're only sequencing the assay product itself, and you're not simply shotgun fragment sequencing all input material that could be present in the assay. So the algorithm simply counts reads that match each of the probes that were present in the assay, whether it be the whole transcriptome or smaller targeted panels. The algorithm is tolerant to mismatches that represent sequencing errors. The identity of each of the probes are associated with the gene targeted, and then a table is output in a CSV format that's essentially a matrix of all of your samples along the top and all of the genes present along the vertical and the number of counts filling out the matrix. This is a very fast analysis. It takes a matter of minutes to be performed on a standard CPU. It does not require complicated statistical deconvolution to identify isoforms or disambiguate multi-mapping to multiple similar regions in the transcriptome. This is because the bioinformatics is front-loaded in the assay, in that the probes that we design are carefully identified through a proprietary pipeline that ensures that the reads coming out of the assay are unambiguously assigned, and don't require then a bioinformatics team later on to unravel this data. It greatly speeds up the process as well as takes away a lot of sources of variability and potential error. With the CSV table that is generated, downstream analysis is possible through a number of approaches. BioSpyder offers TempO Seq software, which is a cloud-based tool for the initial steps of visualization and analysis through a number of approaches, including PCA, clustering dendrograms, and differential expression. Furthermore, the file output is a standard format that can be used by a variety of standard tools that may be more specifically geared to your application of interest. The important point to make through all of this is that a key advantage is the efficiency which is gained through the sequencer. Not only is the assay very easy to run in high throughput, as I've shared, it also is very high throughput on the sequencer. In general, all things being equal, TempO Seq only requires about 10% of the reads for the same quality that you would expect from other sequencing-based assays. This translates to the case, just some examples demonstrated in this table, where with the whole transcriptome, you're able to run tens to hundreds of samples on the low and medium output sequencers from Illumina. Moving to a high-seq obviously allows you to run many more samples. When you perform the targeted assays, in the case of hundreds or thousands of genes in a panel, rather than the whole transcriptome, which is 20 to 30,000 genes depending on the species, you're able to now increase your throughput even further and therefore reduce the cost per sample of sequencing. It's important to point out that the sequencer in this case is really just counting the barcodes that are output from the TempO Seq assay. And so sequencing errors become insignificant because, as I said, we know the input sequence is going in, and so sequencing errors can be unambiguously identified rather than being a source of statistical noise. So with that, we offer a variety of these kits from the whole transcriptome in human, mouse, and rat, the surrogate transcriptome, which represents a pathway-based approach that covers a variety of important genes in addition to capturing most of the content, the information content in the transcriptome, but with many fewer genes. You can also create custom panels based on your own gene list of interest and combine that, if you wish, with this surrogate assay to also provide that broad pathway-based analysis. The surrogate is available for human, mouse, rat, and zebrafish, and custom gene panels can be created from essentially any source, whether it's database sourced information or simply FASTA sequence that you have generated or access to in other ways. So with that, I'll end this introduction. Please, as I said, if you have further questions, check out our website or send us an email. Thank you very much.